Hello, welcome to another screencast where this time we'll be discussing the large intestine and what's going on in the large intestine, which is also sometimes referred to as the colon. Okay, so large intestine, colon, same difference here. So by the end of this, hopefully you're able to identify and describe some of the gross anatomical features and the subdivisions of the large intestine. Two here, the microscopic anatomy of the mucosa, right? So similar to the small intestine. The digestive processes of the large intestine, and then we'll discuss here the difference in the motilities here of segmentation, peristalsis, mass movements. We'll highlight some of down here the explanation of the role of bacteria flora in digestion, and then how mass movements are associated with defecation. Right? And we'll deal with some of the regulation of this in another screencast. So here's the large intestine, or the colon. Again, this is one-way flow or propulsion here. We're going to start at the tail end of the small intestine right here with the ileum. And so this is rightly here small intestine, and the small intestine is going to join the large intestine here at the ileocecal valve. Ileo, of course, for ileum, and then cecal for the cecum, which is this area, this pocketed area, sort of right here. So this is the cecum. An extension of the cecum then is this vermiform appendix, and that appendix may be anterior, maybe posterior, inferior, hanging down there, so that varies. And this appendix right here. The cecum and the appendix house a lot of the bacteria, right, which we refer to as the flora, right, of the large intestine or the gut. So gut flora, if you hear this, right, they're talking about the bacteria in there. An example of, there are many, many bacteria in here, but one example of them is one that you probably have heard of, and that's E. coli. So this is short for Escherichia, and in coli, so the species name here is because these bacteria live in the colon, right? So hence this is E. coli. Now these bacteria are not harmful if they're outside of the gut. They're not harmful inside the gut because they are playing major roles in digesting, and they make available many vitamins. Right? So make many vitamins available, right, through their metabolic processes, right? And especially the B vitamins and vitamin K. Right? So a lot of their digestive processes, a lot of the metabolic processes, one of the byproducts of that is they make the B vitamins and vitamin K. Right? So as food enters through this ileocecal valve, actually it's not food anymore, now it is chyme. Right? And so as this chyme comes in, it's regulated by this ileocecal valve, so it doesn't all just come in at once, right? The ileocecal valve is going to open, close, and sort of regulate the movement of chyme into the cecum there. Once that chyme enters the cecum, it's then going to proceed up the ascending colon, right? And then it's going to turn here at the hepatic flexure, hepatic liver. So the liver would be right here. So this flexure is the turn right there. Right? And then the stuff will enter the transverse colon, or going across the top here, the superior part. It's in the ascending colon and the transverse colon where any absorption and secretion is going to take place. The major role of the large intestine is that it will be absorbing water and also sodium ions, and will be secreting potassium ions. There isn't hardly any absorption of carbohydrates, proteins, lipids here, because that's already all been absorbed, or whatever's been absorbed is going to have been absorbed back up here in the ileum and the jejunum. And so this is all now undigested material, right? And that has made it past the small intestine. But what will be absorbed, so what's going to come out of the lumen into the bloodstream, is going to be sodium ions, and then following that will be water. And this is up to about, on average, 2 liters of water a day are reabsorbed into the bloodstream from the from the chyme that's in the large intestine there. So this is going on in the ascending colon and the transverse colon. The other thing that is being absorbed are going to be the vitamins. So many vitamins get absorbed here. This looks like vitamin S. Vitamins there, and as well as some drugs or medications will also be absorbed here, which is the reason why some medications are administered rectally, right, in the form of a suppository. So you put the drug in through the anal canal here into the rectum, and then it's going to be broken down, and then those drugs or medications are absorbed across the large intestinal wall there. So as we proceed through, as the chyme proceeds through here, it's being dehydrated because water is going out. And so by the time the chyme here makes this downward turn at the splenic flexure, right, the spleen would be right here, it enters the descending colon, and then we'll enter the sigmoid colon. Sigmoid is S-shaped here. It's the descending colon and the sigmoid colon here that are mainly for storage of feces. Right? And so the feces has been dehydrated here. If the feces is overly dehydrated, then you become constipated. And if it's not been dehydrated enough, then you experience diarrhea. Right? So just the right amount here makes the feces sort of compacted, and then it will pass into the sigmoid colon. Right? From the sigmoid colon, then, the feces enters the rectum, and by the time the feces enters the rectum, this rectum is distending, right? And we know what distension causes. Distension will signal muscular contractions, right? And then you will have defecation. Right? This anal canal here is, there's two layers of smooth muscle here. There's an internal sphincter, and then there's an external anal sphincter here, and they will relax, and then the feces will be voided, right, through the process of defecation. So there's a quick tour through the large intestine. If you look at the surface of the colon, it has these little pockets here, and these are the haustra. Right? It has this ribbon, which is this is ribbon here. The Coli. And it's just a piece of smooth muscle that is extended down there. And then you have these epiploic appendages, and these are fat deposits. Right? And their presence is relative mystery still. Like, we don't exactly know why those are there. So let's take a look here just at the section. So let's go down to the microscopic level here. We see that those haustra, so again, here's the Coli, right? These haustra are these little pockets, right? And this is all muscular tissue here. Right? And the little pockets are places for segmentation to occur. So this is all about the motility here. This is from the coloring book. So the motility, the movement of the chyme through there gets segmented, and these little haustra, the chyme gets sort of stuck in here, and the haustra will churn that, and that's what we see here in this segmentation process, right? Then, so this is one form of motility. The second form of motility here is the peristalsis, which is this rhythmic contraction, and this happens at a much slower rate. And so these rhythmic contractions, every hour or so, you get these movements, right, pushing that chyme through. This is happening a little bit more constantly. Right? Every once in a while, you get this peristaltic wave moving through, which then loads the feces into the descending colon and into the sigmoid colon. And once you get that compacted enough here, you end up getting this mass movement, right, which is one very large contraction of the smooth muscle there in the descending colon and the sigmoid colon and the rectum, and then you have defecation or voiding of the feces at the other end. So three forms of motility there: the segmentation, the peristalsis, and then mass movements. Right? Microscopic level. The section through here is like a section through any of the rest of the alimentary canal. Let's start on the outside. You have the serous membrane. This would be the visceral peritoneum. Then you have longitudinal and circular muscular layers here that are responsible for that segmentation, for the peristalsis, and for the mass movements. There's a submucosa, and in the submucosa you have the lymphatic nodules, where you're going to have immune system cells, immune cells hanging out here, so that in case any nasty viruses or bacteria sort of cross the mucous membrane here, the lymphatic system is ready to detect that, or the immune system rather is ready to detect that and set off an immune response. Then you finally have the epithelium, and this epithelium is a simple columnar epithelium, and there are these crypts, or these are the ducts opening there. And so if we look at this, there are much fewer of them. So these are not villi; these are larger scale than villi would be in the small intestine. And the simple columnar epithelia that are there do not have microvilli. So if we look over here, here's the epithelial cell. Right? So absorption will be going across these epithelial cells, and you're going to be absorbing, but then you'll also be secreting, and that is the job of these goblet cells here, and so those are in blue. Right? And this crypt is the duct, so any secretions are going to go into this duct and then be secreted out this way, but most of your absorption, again, water goes across here into the bloodstream, and sodium ions are going to go through here as well. Right? So there's some of the microscopic anatomy, some gross macroscopic anatomy. One final thing here, let's talk about just real quick the bacteria that are in the gut and feces itself. So feces is undigested and unabsorbed nutrients, and also bacterial waste and bacteria. So if you were to take a fecal sample and look at it, about 1/3 of it would be bacterial waste and just bacteria itself. So by the time that feces comes out, it has changed. Right? So let's look at at point 1 versus point 2 over here. So if we look at the chyme that's here versus the chyme that's over here, at point 1, most of the undigested material that enters the chyme there is plant material made up primarily of cellulose. This is undigested. We don't have enzymes to digest cellulose, right? So the cellulose concentrations are relatively high. The bacteria concentrations here, right, at point 1 are relatively low. But as we move from point 1 to point 2, right, the cellulose concentration is almost completely gone, right? But the bacteria numbers, the bacteria levels here are really high. So what's this tell you? This tells you that the bacteria are consuming the cellulose, right, and using it to power reproductive efforts here. And so as you move through, the bacteria are metabolizing. The bacteria metabolize cellulose. And when they metabolize cellulose, the byproducts of that, again, are the B vitamins, right, vitamin K, and other substances that the body will need. Right? But you're making more and more of these bacteria. These vitamin B and other substances, again, get absorbed out here. But you're making more and more bacteria, and so those bacteria make other metabolic wastes. Right? Metabolic wastes. Waste products, and you have bacteria dying in massive amounts here, so that by the time you reach point 2 over here, again, about 1/3 of this is bacteria itself or bacterial waste products, right? And then the other 2/3 is anything else that was not digestible, right? That was not broken down by any of your own enzymes or the enzymes of the bacteria in there, right? So there's a significant research going on right now into the gut flora and health and well-being and the correct levels of gut flora and things like that, and we can discuss that a little bit more in class. Right? So I think like I said, I'll wait for the regulation here in another screencast, but you should be able now to summarize here, talk about the gross anatomical features and then the subdivisions of the large intestine, the microscopic anatomy, right, at the mucosa level, the digestive processes, so what is digested, what's not, what's absorbed, right? What's secreted, right, along the way, and then the difference between segmentation, peristalsis, mass movements. A little bit about here the bacterial flora, right? And then how the mass movements bring about defecation. Again, we'll deal with this when we talk about the regulation of processes in the large intestine. Okay, thanks for listening, and bring your questions to class. Whoops.